Hello, in this podcast we're going to be learning about the cell membrane, or another name for it is plasma membrane. I'll be using those two terms interchangeably. And in particular, we'll be learning about how substances cross the cell membrane. Now here are some things about the plasma membrane that you need to know. Some things we've already covered. First of all, it is the border of the cell. If you're on one side of the plasma membrane, you are inside the cell. If you're on the other side of the cell membrane, then you're outside of the cell. And among its jobs are that it keeps what should be inside the cell inside, and what should be outside the cell is kept outside. And also it regulates what passes through it. And now this has to do with maintaining homeostasis, both in the cell and throughout the whole organism. Like, for example, throughout the whole organism would be like, let's say you've just eaten a meal, and you have digested the food, and that food is leaving your small intestines and it's entered into the blood. So you have much more sugar in your blood than you are really than it's healthy for you to have. So what happens is that your pancreas, some of the cells in your pancreas, will allow a hormone called insulin to pass through the plasma membrane, and this hormone will then go to your liver, and it will tell your liver cells to actively pump the sugar molecules into the cells in the liver, and that will lower your blood sugar content. And someone who's a diabetic is either unable to make the insulin, in the case of someone who has type 1 diabetes, and someone who has type 2 diabetes, then the plasma membranes of those cells don't really detect the insulin as well as they used to. And also, by regulating what passes through, it also protects the cell by keeping things that should be out of the cell, and what's in the cell. Some of these life processes that are covered is regulation. We just went over the regulation. Nutrition, by bringing food from outside of the cell into the cell. Excretion, by letting the wastes that are in the cell out of the cell. And also transport, by just moving things around. Now here's the basic structure of the plasma membrane. It is a phospholipid bilayer. Now if you just think in terms of, like, imagine that you've got a salad dressing. Salad dressings are made of something that is mostly water, that's water soluble, and also an oil. And you notice how you always have to shake up the salad dressing, because the oil and the water will separate, because those things don't mix. So you can take many substances and divide them into two categories: substances that are water soluble and substances that are fat soluble. An oil is a type of fat. Now now most of the stuff that's in our body and what cells are found in has a lot of water, so it's water soluble. So what the plasma membrane does is that it makes its barrier fat soluble, so that most substances which are water soluble can't get through it, because they can't dissolve their way through it, just like the water can't go through the oil. So you've got a phospholipid bilayer where the edges of the plasma membrane are water soluble, and that allows them to contact the water inside the cell and outside the cell, and the inside of it is fat soluble. So that means that only fat soluble substances can pass through the plasma membrane unaided. Those are the only things which is not a barrier to it. And some substances like that would be things like steroids, and that's why steroids are so dangerous, because the cell membranes just can't stop them from going through it. Now there are 4 ways that substances can pass through the cell membrane. And before we get to those 4 ways, we have to first talk about the permeability of the cell membrane. And permeability is the degree to which a barrier allows substances through it. And we'll be doing a state lab in the spring on permeability and also on diffusion. And if something is permeable, that means that anything can pass through it. And if something is impermeable, that means that nothing can pass through it. Semi-permeable means that there are some substances that can pass through it. Like, for example, let's say you have, say, some filter paper, and you take some hot water, you mix it in with tea leaves, and then you pour that stuff through the filter paper, and the liquid the tea will go through the filter paper and into your cup, but the tea leaves will stay behind. So there, the filter paper is semi-permeable, and it's by size. The small substances can go through, larger substances cannot. And then there's selectively permeable, which means that the barrier, in a sense, can choose what things go through it. Now, the cell membrane is semi-permeable and selectively permeable. Only certain things can go through. Sometimes certain things it can't choose, like water is just able to go through, and other things are selectively permeable, like sometimes it will let certain things through, and sometimes it won't let those same things through, if the conditions are different. Okay, so now here's the first way that substances can pass through. It's called diffusion. And diffusion is going from high concentration to low concentration. That means where you have a lot of something to a little bit of something. It uses no energy, and the cell is not able to control the diffusion. Now here's an animation that I want to show you about how diffusion works. So first thing is that molecules are in constant random motion, and one result of this motion is that dissolved molecules become evenly distributed throughout the solution. The tendency of molecules to spread out is an example of diffusion. Let's say that here we have a lump of sugar, and here, because they're all very close together, that means that they're very, very concentrated. And we're going to see how this lump of sugar can, through diffusion, become evenly distributed. So it begins to dissolve, and you see that it's dissolved, but they're all really close together, and the sugar molecules are randomly moving around, and just through that random motion, they just tend to go from where you have a lot of them to where you just have a little of them. So they're going from an area of higher concentration to lower concentration, and that just simply is diffusion. And you don't just simply see this, you see this throughout your life, not just in biology, not just in this. Like if you are cooking something, the particles that make the odor of, say, your cake that's baking or whatever food you're making spread throughout the whole room and your whole home is diffusion. Osmosis is the second method, and osmosis is diffusion. Only thing is that it's diffusion of water, so in a sense it's a special case. The best way to think of it is that instead of looking at the concentration of, say, the sugar in the water, you're looking at the concentration of just the water. If you have a lot of sugar in it, that means you have a little bit of water. If you have a little bit of sugar, that means you have a lot of water. And this diagram that you see here is one that was taken from a Regents exam. And this shows basically what happens to a substance due to osmosis. And we'll be seeing this in the lab very shortly. And basically, look on the top there, you see that you have a solution that is 95% water and 5% starch, and it's in a bag that is semi-permeable. It will let the water through, but it won't let the starch through. And if you put it into a beaker where the water is 100%, that means that the water is going to diffuse into the cell because it goes from higher concentration of 100 to lower of 95. And as the water goes into that bag, because there's more water in the bag is going to expand. And if you imagine that being a cell, that's the same thing that would happen to a cell. On the other hand, if you're taking the bag and you're putting it into a solution which has exactly the same concentration of water, then you won't have osmosis, and this because there's no concentration gradient, and the size of the bag won't change. So if you put it into a solution where there's a lower concentration of water outside of the bag or the cell, then the water is going to diffuse out of the cell, and the cell is going to shrink, like you see happening to the bag here. Third method is called facilitated diffusion. And if you remember from Spanish, fácil is the word that means easy. So facilitated diffusion is diffusion that's made easy. There's something blocking the diffusion. And what happens in this case is that the substances are water soluble, so they can't go through the cell membrane, but there are protein channels that are able to let them through. When they let them through, that's facilitated diffusion because it's aiding, the protein channels are aiding the diffusion. This allows the cell to control what goes through, so that those channels only let certain substances through. That's a measure of control. And also, it'll only let those substances through when it needs it to go through. Other than that, facilitated diffusion is just like diffusion. It's high concentration to low concentration, and no energy is involved in letting this happen. The fourth kind is active transport. And here using the same protein channels in many cases, but it's going in the opposite direction. It's going from low concentration to high concentration. So it's the opposite of what we've seen so far. And another way it's the opposite is because it's going against the concentration gradient, that uses energy, and the energy is in the form of ATP. And the way you can remember that is this, is to think of the word active. Active means that it has to actively be involved. The cell's actively pumping the substances across the plasma membrane. Now I'm going to show you some demonstrations that will help clarify these types of transport across the cell membrane.
Here I'm going to show you an analogy to show you how to help you understand how substances cross the cell membrane. Think in terms of a ramp. Now let's pretend that the cell membrane is right across here, across the center of the ramp. And substances will move depending on the concentration gradient. And where the ramp is high, think of the concentration as being high. Where the ramp is low, think of it as being low. Diffusion goes from high concentration to low concentration. So that means that the marble goes here, and down it goes. It took no energy to do that. That's diffusion. You go from high concentration to low concentration. Facilitated diffusion, what happens is that there's a gate in the cell membrane. Let's say that let's say this fork here is the gate. And in order to, so it can't get through unless it's got the key to open up the gate, and then the gate opens, and down it goes. That's facilitated diffusion. Active transport is going from low to high. So if I just so it won't, in order to get this to go up, I have to push it up to the top of the ramp. And hopefully it won't fall off the ramp. And that's active transport, going from low concentration to high concentration. And since it's going up the ramp, that requires energy. Now the speed that it will work, that depends on the difference between between the high concentration and the low concentration. Like if I were to lower the ramp, now the concentration gradient is much smaller. There's not as much of a difference between the high concentration and the low concentration. So it goes down a lot slower. And that's also how the ramp can help you. So the bigger the difference between concentration on one part of the cell membrane to the other part of the cell membrane, bigger the difference, the faster it's going to go. And when it's going from high concentration to low concentration, it doesn't require energy because you're going down the ramp. When you're going from low concentration to high concentration, like an active transport, going up the ramp, and that requires energy.
Now we come to the rest of the structure. We've seen before that we have the phospholipid bilayer. Also in the cell membrane we have proteins that are floating in it. And some of these proteins are receptor molecules. And receptor molecule is an important vocab word. And we're going to run into it several times over the course of the year. And these are basically substances that are used to, the receptor molecules are used to identify substances. For example, take the hormone we talked about before, insulin. Insulin goes throughout the whole body, but it only needs to get into certain cells, and the receptor molecules will bind to the insulin and will identify it and let it through. And if there's cells where the insulin isn't supposed to affect it at all, then those cell membranes won't have the receptor molecule, and the cell will basically just ignore the insulin, and the insulin won't go into the cell at all. Another thing the receptor molecules are used it's sort of like a name tag for the cell. The cells have, those name tags will identify the cell as you, and that way your immune system will see that name tag and won't attack your cell. If those name tags are not there, if those receptor molecules are not there, the cell membrane will say, this is an invader, and it will attack it. And we'll see more of that when we get into the immune system. And also you'll find in there protein channels, in the cell membrane, and these are proteins that allow the substances to cross the cell membrane, and that would either be facilitated diffusion or active transport. Before I move on, I just wanted to point out with the receptor molecules, the way the receptor molecules are able to identify what goes in there is purely by shape. It's just like a lock and a key. If the receptor molecule is like the lock, and if the substance is able to fit into the lock like a key into a lock, then the receptor molecule identified, and if not, then it won't say let it through the protein channel. Now we come to our concluding questions, and once again you need to answer these questions and hand them in to me tomorrow. Number 1, what is the plasma membrane's jobs? Number 2, which type or types of transport are from high concentration to low concentration? So it's which ones of those 4 types, and also which of those 4 types of transport require energy? How do receptor molecules make the cells semi-permeable? Or make the cells selectively permeable, in choosing what goes through the cell membrane? And what is the role of protein channels?